So I'd like to start by telling you a story that happened a couple of years ago in 2015 in Angola. There were a few cases of yellow fever reported, which is a disease that's transmitted by mosquitoes. It's usually spreading in monkeys in the jungle, but once in a while it jumps into human populations. And we usually pay attention because it's a pretty nasty disease. It'll give you fever, rash, nausea, jaundice, and can even kill you. And sometimes it'll just trickle out, and sometimes it becomes a full-blown outbreak. And in this case, within less than a year, there were 7,300 suspected cases throughout almost every district of Angola, and it even spread into the neighboring country of Democratic Republic of Congo. Now, at this point, the World Health Organization and a number of other groups were really concerned. And the reason for that is that once you look at travel maps, within about 48 hours, over 1 billion people could be affected by this disease. So this is a global pandemic threat at this point. So what all the epidemiologists and ecologists and health professionals did was they built some ecological models and figured out how many vaccines did they need to get a handle on and distribute, where did those vaccines need to go, and in what time frame did they need to be delivered. And based on that, they were able to deliver over 19 million doses of vaccines to the right people, the right places, and critically before the rainy season starts, which is when the mosquito population will explode. And with that successful campaign, the last case was reported in June 2016. And this is probably why you never even heard of this outbreak, because it was contained to these two countries. Now, in this example, the reason this was even possible is because we know so much about yellow fever. We know the ecology and the behavior of the vector, we have a vaccine, and we know how it's spread through people. And this is really the motivation behind a lot of my work. I'm interested in what are the ecological conditions that allow diseases to emerge in the first place, and how can we use those relationships to predict outbreaks in the future to either slow or stop their spread, or ideally to prevent them in the first place. And the way I do that is by asking a couple different questions. For instance, what makes certain individuals or populations more susceptible to infection? How fast does disease spread? Can we forecast outbreak risk in the near future? And how will global change in the form of climate change, land use change, and urbanization shift the burden and distribution of diseases in the longer term future? And so I asked these questions on a variety of systems. A lot of them are marine, such as clams, coral diseases, sea star diseases, as well as a variety of mosquito-borne diseases that affect people. And while they may seem like really disparate ecosystems, there are some really common threads that I find very exciting and interesting. The first is that for all of these diseases, climate is really important, either for the host, the pathogen, the vector, or some combination of those three. These diseases tend to be habitually underdetected, and there's no widely available treatment for any of the diseases I study, which means predicting outbreaks is even that much more important because it gives us an opportunity and some time to do something about mitigating the effects. So today I'm going to tell you two stories about some of the coral disease research that I've done and the mosquito-borne disease research that I've done. And since we're at the Center of Excellence for Coral Reef Studies, I'm going to focus on my coral disease research first. So in coral disease studies and coral health in general, one of the big challenges is that there's really low background levels of disease. Outbreaks are really patchily distributed in space and time. And most specifically important to coral is that these diseases are happening across these really expansive ecosystems. And so without a really good monitoring or surveillance system in place, like we have for humans, it's really hard to even detect them. And how can we predict or do something about them without knowing where they are in the first place? And so in order to study this, we first need to capture a few outbreaks and understand the dynamics. And I had one such opportunity during my PhD, which I'll tell you about now. So now I'm bringing you to Kaneohe Bay in Oahu in the Hawaiian archipelago. This here in the middle is the Hawaii Institute of Marine Biology. That's where my lab was based during my PhD. And every day to get to work, we take a boat from this pier over to the island. And one day the boat driver, Bobby, said he's been noticing a lot of really white coral. We've all heard this before. However, it was winter, it was cold, and we just had a big rainstorm. So the probability that it was coral bleaching was pretty low. So a number of students and faculty went out and explored, and it turned out to be this mystery disease that we had never seen before. It was exclusively affecting one species of coral, Mantipora capitata. It caused this rapid tissue loss where the tissue would dissolve and disassociate from the coral skeleton. It also manifested in quite an unusual way from other tissue loss diseases or white syndromes that you may have heard about in terms of how it spread on the colony itself and where we were finding it in the bay. And later we found out from histological investigation that it was a completely new disease that had never been heard of before, which was unfortunately termed colony-wide protein-losing cytopathology, which I'm not going to say again. I'll call it tissue loss disease, but if you want to learn more, you can look it up. So what we initially found out is that the outbreak was really located in the center of the bay, with the epicenter where this big red dot is. So what I decided to do is I wanted to capture the full outbreak cycle, and so I selected 3 reefs that were just adjacent to the epicenter, thinking that would be the next place it spreads if it continues in the pattern where it's gone, moving up the bay. And so myself and my colleagues asked a few questions. The first, could we determine how quickly this disease spread? And what factors were increasing or decreasing any individual colony's disease risk? So how did we do this? How do you estimate disease transmission in a coral reef when you're swimming around? Well, what we did was identify 200 visually healthy colonies, and we monitored their health state every day for 5 weeks. In addition, we collected information on a number of both colony host traits and then at the community scale that we hypothesized could be associated with disease risk. So the first of which was colony size, because for lots of diseases, the animal size really matters in terms of its infection likelihood. We also observed depth, position on reef, so reef crest, slope, flat, and sedimentation on the colony, which are all proxies of temperature, irradiance, and water flow. And then we also looked at community composition, so the density of Mantipora capitata, the host, which can be important for how many colonies can actually get infected, coral cover and coral diversity. So there's some interesting hypotheses about how the composition of the community can actually influence the risk of disease to other individuals. So if you have a lot of host species that can't get infected, that they can act as a barrier, let's say, for disease spread. And then disease pressure. So these were measures of the severity, the distance, and the size of the nearest neighbors to each colony that we were looking at and their health state. So this is a map where all of the little circles are the colonies that we monitored on the 3 reefs. And I'm going to show you a series of maps here about how the outbreak progressed. And so when the colony is susceptible, it's gray, so it's never been infected. When it's red, it's gotten infected. And when it turns black, it's either died or recovered from the disease. And so what you'll start to see is dots randomly turning red, then some dots close by will turn red, indicating more colonies getting sick. And then you'll start to see ones turning black in a similar pattern to how they got infected in the first place as they start to recover. So this is visualizing the outbreak spatially. We can also look at it over time. So here on the x-axis is days of monitoring, and the y-axis is the number of individuals. So the gray line is the susceptible, where we start all 200 colonies were susceptible to disease. As we go through the outbreak, a little over 50% of the colonies got infected. Early on, we see the red, the infected colonies increasing and then decreasing. And as you have the decrease in the infected colonies, the removed colonies increase. So at every point on this line, all 200 colonies are accounted for. Now, for any disease ecologists in the room, this is a very typical plot. But what's atypical is that we almost always see these 2 lines overlapping, the susceptible and infected. And the reason for that is because usually an outbreak stops or burns out because you run out of susceptible individuals to get sick. So this was really surprising, and one hypothesis we had is that this big rainfall event triggered the outbreak. Maybe there was an influx of pathogens from the streams and rivers coming out to sea, and then it abated. There was no more rain, and so the pathogens disappeared and colonies recovered, and that was the end of it. So that's one possibility for why we didn't see the expected pattern over the outbreak. What was really exciting to me is this was one of the first times we've ever actually captured the rate of spread of a disease in coral. And what this is showing is essentially the per capita infection rate over time. And it was actually much slower than we expected given the initial outbreak in the middle of the bay. So at the peak, somewhere between 2 and 2.5% of colonies would get infected per day. Now, even though that's slow, background rates are so low that this is exceptionally high for a coral disease. Normally for a tissue loss disease, you might see 1 in 100,000, maybe 10,000. So the order of magnitude is a huge difference during this outbreak compared to what you normally see. The second metric we were able to capture is R effective, which is essentially how many colonies will each colony get sick. And so if you're at Ben Volker's talk last week, he talked about this a bit. When you're at 1, it means every colony infects at least, or just one other colony, so the outbreak will be sustained. If you have fewer than 1 colony diseased by every colony, the outbreak will die out, and if you have more, you'll have exponential growth. So what we ended up seeing, how this changed through time, well, on average, it was a little less than 1 colony getting infected by every colony. And there were really only 6 days where the outbreak was growing. And so we think the reason for this, even though we saw an exceptional spread of disease in the first place, is that we actually captured the second wave of the outbreak after the initial one that I showed you initially. So we also found some interesting factors associated with any colony getting infected. And the first is that we found larger colonies were more likely to get sick. There's 2 hypotheses for why this could be. One is the surface area hypothesis. So you have a larger colony, there's more surface area exposed to any pathogens in the water column. The second hypothesis is the age hypothesis. Although not all the time, in general, larger colonies tend to be older, so they could have a reduced immune system or been exposed to more chronic stresses for longer. So if the surface area hypothesis would explain why we're seeing larger colonies getting sick, we'd expect to see multiple lesions anywhere around the colony and in proportion to the size of the colony. If we thought it was the age hypothesis, we'd expect to see that the disease lesions would start in the oldest tissue at the base of the colony and spread outwards. And that's exactly what we saw. So we believe this is the mechanism for why larger colonies were getting infected, although we weren't able to actually tease apart whether it had to do with exposure or immune capacity. We also found that colonies that were located near more diseased colonies were more likely to be diseased. That's not so surprising, but we were actually able to quantify that. So depending on the size of the disease lesion, a colony was 1.2 to 1.6 times more likely to get infected than the average colony. And then the third thing we did was look within boundaries at increasing distance from each focal colony. And what we found is that transmission was greatest within just 15 meters. So this happened to be a disease that was driven by a highly localized disease spread. So for every colony, what mattered most about its transmission risk was whether the colonies within a 15-meter boundary were also diseased. And so when we look back at this previous plot that didn't overlap, and then we overlay the proportion of susceptible colonies within that 15-meter boundary, we actually see the overlap exactly where we would expect. Now it's important to point out the second y-axis. This doesn't actually go to 0, but this is a subsample of the population, and I think the timing matches up so perfectly that it really shows how localized the community mattered in terms of infection risk. So what I told you about was a single outbreak event in a very small location on a couple patch reefs. And it was an incredibly interesting opportunity to study disease. But it was really only possible because a number of fortuitous events happened. Somebody noticed it. There was a large group of researchers there with boats and scuba gear and plenty of time to investigate this. This isn't really practical on the scale of coral reefs and coral diseases. So this is a map of where coral diseases have been reported. And all I really want you to take away is they're pretty much everywhere that coral reefs are. And so this is really a global issue, and it's important to think about, is it possible and how could we scale this type of research up? And so that's the type of work I'm doing now with a project called 4C. We're developing forecasting models of coral disease risk throughout all the regions highlighted in pink, throughout the Indo-Pacific Ocean. And one aspect of this is putting together an outbreak response network in different areas. And we have had one outbreak since this has started. We've had collaborators in Guam who caught it, and we're on the ground to collect more data. But for the most part, we're focused on using existing data sets to understand the drivers and forecast outbreak risk. And we're doing that for 3 widespread diseases throughout the Pacific. One is tissue loss, like I just talked about, diseases that spread throughout the colony, killing tissue, and these can be chronic or acute, but when they're acute, they can kill colonies in less than a week or 2. So it happens really dramatically, and it can have a big effect, even if it's pretty localized. We're also looking at black band disease, which is a seasonal disease that affects lots of different coral species. And then growth anomalies, which is more like a coral cancer. It tends to not kill colonies, but it does reduce growth and fecundity. So it can have longer term effects on reef trajectories. And while we're using a suite of drivers in these models, things like wave energy, irradiance, coral community composition, the 2 that we're focusing on most strongly are temperature and water quality. And the reason for this is based on previous studies. There's a lot of pretty strong links to different aspects of disease, as well as the fact that these 2 metrics can be remotely monitored. So it means we can nearly continuously monitor a great expanse of ocean continuously through time and continue to reduce outbreak risk. So what we've found so far is some interesting relationships with temperature. Unlike coral bleaching, it's not just anomalously warm temperature, but anomalously cold temperature as well can really affect disease dynamics. And that's because it affects both pathogen survival, growth, and ability to infect coral, while simultaneously affecting the coral susceptibility to infection. Similarly, for water quality, and here we're really focusing in on chlorophyll and turbidity, we know that it can influence both pathogen growth and transport, specifically when you have rainfall events from high islands with rivers and streams that flow into the ocean near reefs, while at the same time affecting both coral susceptibility and potentially coral feeding. So in certain conditions, corals are more likely to heterotrophically feed, so pull food out of the water column, and that will increase contact rates with other particles that may be carrying pathogens or pathogens themselves. And so based on some previous work from Scott Heron, who's at JCU, and myself, there's experimental disease risk products currently available on NOAA's Coral Reef Watch website. These are just based on temperature, but they're producing outbreak risks at certain points in time every year. What our project is doing is a complete overhaul of these models. We're expanding it to the 3 diseases I told you about and across all the regions on that map. And there'll be continuous outbreak risk alerts, analogous to the bleaching alerts that will be available on their website. And all the virtual stations that they use within our study region will also have outbreak risk, icons that you can click on. But what we're really excited is what you'll be able to do is click on those icons and be taken out of the NOAA site into a separate explorer. And what we'll have here is a seasonal outlook for a disease risk, which is about 6 months prior to when you'd expect an outbreak, a 4-month forecast, and a near real-time forecast. In this explorer, you'll be able to toggle between those maps You'll be able to look at the uncertainty in our estimates and look at the different diseases alone or together, as well as have the capacity to change certain layers. So let's say you're a manager at a reef and you want to address disease or maybe overall health, and you think, what if I were able to reduce resuspended sediment by 10%? What might that do to the risk of tissue loss? So we're really excited about this, and there should be, fingers crossed, a beta rollout of it at ICRS in July 2020. So definitely stay tuned if you're interested. We got a lot of great feedback from different researchers and user groups in locations that this is the type of output that would be useful. But we will be looking for beta users, so if you're interested in checking it out before, please let me know. So that's somewhat of an overview of my coral disease work. And now I'm going to shift about 180 degrees and talk about mosquitoes. And I know this is the Center for Excellence for Coral Reef Studies, but I was really excited to talk about this research because Australia, and Caldwell in particular, is a very cool place for mosquito research. So I don't know if you know, but mosquitoes are the deadliest animal in the world. They kill more people, and only about 5 species, than all other animals combined. And Caldwell is really at the forefront of changing the game in dengue transmission. Dengue is one of the diseases that I focus on that mosquitoes spread. It's often referred to as break bone fever if you haven't heard of it. It's pretty nasty. And they're spread by these mosquitoes, the species called Aedes aegypti. They're here in Australia, and if you were here in 2014, you might be familiar with the release of 4 million additional mosquitoes. And as much as that sounds crazy, the reason is because there's this naturally occurring bacteria in a lot of insects called Wolbachia, and if you infect it into the mosquitoes, it can actually disrupt the virus cycle in the mosquitoes and reduces transmission. And so this is one of the few places in the world where these have actually been released, and there's quantitative data to show that it's working. It's pretty exciting. So this is the increase in mosquitoes with Wolbachia. This is the number of dengue cases in 2014. You're no longer seeing local transmission of the disease. And here is Wolbachia coverage spreading through space. This is actually amazing and a little hard to believe. They only travel about 150 meters, so the fact that this is saying they've spread 70 kilometers is shocking. But yeah, so this is an exciting place, and I thought I would just share a little bit about the other type of research that I do. So traditionally, the way that people thought about how mosquitoes spread diseases is that they're basically flying syringes, and they'll just infect anyone with no plan or action. They're not animals, they have no mind. But that's really ignoring all the important ecology and behavior that we know is important to mosquitoes and really should dictate a lot of the transmission dynamics. So what I wanted to do with my colleagues was build a model that really incorporated these traits. And a lot of these traits are strongly driven by temperature. So a lot of you are probably familiar with this curve. I've already seen it in a couple seminars that I've attended here. But in case you're not, it's a temperature-dependent trait-based curve. So what that means is on the x-axis we have temperature, and the y-axis is performance of any trait. So here you could think of it as fecundity, how much does a mosquito reproduce? And at low temperatures, you have low performance that increases to some optimal temperature, and then you have this steep and sharp decline, and that the performance drops off again. And this is important for every metabolically related trait. So for mosquitoes, that's associated with growth, fecundity, development, the ability of the virus to develop in the mosquito, and the ability of the mosquito to bite people. So it is super important. And we thought if we can incorporate all of these traits mechanistically into a model, we might be able to do a better job at predicting transmission. And so that's exactly what we did, but of course we also wanted to validate that model to see if it said anything about what was happening in real life. So we were fortunate enough to work with groups that have field work already happening in Ecuador and Kenya. And I think these are great places to validate this model because they really, they're basically two ends of the dengue spectrum in my mind. In Kenya, you have very low levels of year-round transmission, whereas in Ecuador, you have these huge seasonal epidemics that die out. In Ecuador, all of the fieldwork was led by Anna Stewart-Agara, and in Kenya by Desiree LeBeau. It's really nice. These programs had 4 sites that we were working in each country across an urban-rural gradient. They also collected mosquitoes and laboratory-confirmed dengue cases over many years. So this is a picture of what the model looks like. Essentially, we split the mosquito population into 3 classes: susceptible mosquitoes, exposed mosquitoes, and infectious mosquitoes, and the human population into susceptible humans, exposed, infectious, and recovered. The solid lines show the direction that individuals can move between the compartments, and the dashed arrows show the directionality of transmission. So for a susceptible human to become exposed, it needs to get bitten by an infectious mosquito. Now, underlying this, we're able to incorporate all of these physiological traits that I mentioned before: fecundity, development rate, mosquito density, biting rate, survival, and virus development. And all of these were based completely on laboratory experiments. So in the lab, people exposed mosquitoes to different temperatures, measured these traits, and all I want you to take away is 1, that it was in the lab, not in the field, and 2, that there are all these unimodal curves. And so each of those traits come into play where I've highlighted in red, just to give you an idea of what that looks like functionally. For biting rate, which affects these 2 arrows, that rate varies with temperature on a given day. We also incorporated relative humidity, which affects how long the mosquitoes live or how quickly they die, and rainfall. Now, rainfall is important for mosquitoes for a lot of reasons. Mosquitoes need fresh water to lay their eggs and to complete most of their life cycle. So you need enough water for them to do that, and not too much water that it's flushing away in the containers that they like to use to breed. So to run this model, we needed some observational data. So we had daily temperature, rainfall, and humidity from all 8 sites. And then we validated it with data. We had trapped mosquitoes at multiple life stages from about 20 houses per site for multiple years. And then, unlike coral, when people get sick, they go to the clinics and the hospitals. So they came right in and gave us their blood and told us they weren't feeling well, and then we were able to test their blood and find virus in their blood. So this is a really rich data set and was pretty exciting to work with. So as an overview, we have temperature, humidity, and rainfall that go into the model. The model is completely based on laboratory experiments. It runs, and the output is predicted mosquitoes and humans in each of these 7 compartments. But what we're really interested in is the total mosquito population through time and the number of predicted disease people through time. So this is an example output. The solid black line is the model predictions, and overlaid is the mosquito abundances tracked at each time point in the circles. So I think this is a pretty amazing starting point, actually. I was blown away that we really were able to take a model based on things we found in the lab, and it would tell us anything about what happened in reality. But at the same time, we wanted to know where the model was diverging and not doing so well. So for each data point, we went back and looked at where was the model doing a pretty good job at predicting mosquito abundance or dengue cases, where was it under-predicting, and where was it over-predicting the number of mosquitoes or dengue cases. And we took all that and put it in classification and regression trees to try and understand some of the conditions that might be driving divergence between what we predicted and what we actually found. And so in this example, if we had very variable rain from day to day in Kenya, we were over-predicting mosquito abundance. And so what we did was take that information and refine our hypotheses about how rainfall was incorporated into the model. We could update the model and rerun it. And so we basically have this iterative model data loop where we were able to refine our hypotheses, specifically about rainfall and humidity, which are far less well studied and understood than the temperature relationships. So I'm just going to highlight some of the most interesting findings. So the first is that we figured out we needed to use different rainfall relationships for Ecuador and Kenya. So this meant there was some context-dependent knowledge that was needed. But it was also counter to what I expected. So this relationship is what I would normally expect with rainfall, which says as you increase rainfall, you'll increase the breeding habitat for mosquitoes until some heavy rainfall where you get flushing. This indicates places where people store water. So in a lot of regions around the world, people don't get piped water into their house, so they have buckets, they leave them, and they use them for washing and cooking and everything else. And so in Kenya specifically, and other places, this has been shown to be a real issue with predicting mosquito-borne diseases because it completely disrupts the natural relationship you would expect. So what I expected was that this would describe Kenya, and this would describe Ecuador. And what we found was the complete opposite. So these are two pictures of Ecuador. It's quite clean and pretty, and for the most part, people don't store water. But what we found out is that even though people get piped water to their home in Ecuador, it's not completely reliable. So people were leaving buckets of water as just in case water and not really paying attention to them. And that's the perfect conditions for mosquitoes to breed because they leave them out. This is what we saw in Kenya. There was trash everywhere, tires and coconut shells, which are known to breed mosquitoes. And so we really expected this should be disrupted by human behavior. However, in the villages we worked in, nobody has piped water. So everybody has buckets, but they use them every day, and they take really good care of them because it's their source of water for everything they need. And so there's no time for mosquitoes to breed, and it's not going to happen even if there was because people are taking really good care of that water. And so this was a super interesting and useful finding and a little bit counter to some of the other larger studies that were just correlating rainfall patterns with dengue cases. So now I'm just going to show a few examples of what the output looked like more generally in the final model where we did incorporate those different rainfall relationships. So these are one site in Ecuador and one site in Kenya. Again, the solid line is the model predictions, and the purple dots and lines are the mosquitoes that were trapped. What we're seeing is pretty good correspondence. I used a measure called root mean squared error, which just, if it's a perfect fit, it would be 0, and it goes up to infinity. So it's quite a good measure in terms of 1.14 and 1.65. We were pretty happy. But I think visually pretty much tells you the story here. However, when we go and look at dengue, it's a little bit of a different story. So here we see a very dynamic map for the predictions, and we only see peaks in dengue incidence once in a while. And what I'd like you to notice is that when we do see dengue incidents, the model does predict there should be dengue. However, the model predicts there should be dengue a lot more than we're actually seeing it. So this didn't necessarily surprise me. There's some really complicated immune dynamics that we completely ignored in humans. And a lot of other things. People have behavior and they travel and they do lots of things that could disrupt our very simple model. But what was really cool, in several of the sites, we were actually able to get data on other diseases that these same mosquito species transmit, which is chikungunya and Zika. And when we overlay those, at every point where we're seeing the mosquito predicting transmission, we are seeing transmission of one of the diseases and sometimes multiple. So basically, our model is doing a really good job of saying when is it suitable for transmission of a virus from this mosquito species, but not necessarily which virus it's going to be transmitting. So yeah, I love the smiles. This is how I felt when I saw this. I was absolutely shocked. So that was very cool. So we're really happy and excited about these results. So going forward, I've been thinking a lot about, now I've been working in really different systems, and what do I as a researcher want to contribute to the field? What do I think is important, and what can I learn from taking these disparate lessons, and how do they fit together? So from more of a methods approach, I'm really interested in combining these correlative top-down approaches that I talked about with the coral studies with these more mechanistic, trait-based bottom-up approaches that we can do with mosquitoes. I think there's a lot of room to improve our understanding of the mechanisms of disease, especially in marine systems, by taking some of these approaches that have been developed in the medical field, which is far ahead just because we've known about them for far longer. And in the same way, how can we bring some of these correlative approaches to improve these mechanistic models? Mechanisms are really good, but they often don't get at those little variations that are important for predicting near future events. And then more broadly, I've been thinking a lot about what are the core concepts that are fundamental to this field of disease ecology, which is relatively young, and what transcends all systems. And so some of the ideas I've been thinking about that I'm pretty excited about, out of my PhD, I think a lot of my work has shown that disease is not quite spreading as far and as fast as we might have expected in the ocean. There's this idea that it should. I think it's quite analogous to larvae in the ocean. There is this idea that they have this long pelagic larval duration and they should spread all over the ocean. And then some geneticists showed us, no, not necessarily. They can stay really close to home. And I think we might have some of the same things here. There's these invisible barriers of currents and eddies and internal waves that might really be dictating where disease can spread in the ocean. And then secondly, in every system I've worked in, temperature keeps coming out as super important, and it's complicated and it's nonlinear and it's super interesting and it's affecting the host and the pathogen, and if there's a vector, it's affecting the vector. And so it's really brought to my attention that this relationship is really context specific and really needs to be thought about carefully, especially when we're predicting out in the future with climate change. So in the early 2000s, late 1990s, there was a couple of key papers that came out and this paradigm that emerged, that a warmer world is a sicker world. And you might have even seen that headline in newspapers. It was in the New York Times and Time magazine. But we think the effects of climate change will be a lot more nuanced, even if you're just looking at temperature. So just to leave you with one example, if we look at a transmission suitability curve across a temperature gradient, it often looks like this. And what this suggests is that how climate change will affect transmission suitability in the future will depend entirely on what the current conditions are today. So if you have a location that's around 16 degrees Celsius and you have a 1 degree increase in temperature, your transmission suitability shouldn't change very much. However, if you're a location around 21 degrees in this graph, that same 1 degree temperature increase will cause a huge increase in transmission. If you're sitting near the optimal conditions, that same temperature difference might not change transmission at all. But what I think is most critical here is places that are sitting on the edge of suitability right now. Because if you increase temperature, you could actually decrease transmission suitability. And so what this suggests is that the burden and the distribution of diseases will not expand and increase everywhere. It will shift and it will change, and different diseases will be taking the niche of other diseases. And so thinking about these mechanisms and how temperature is really driving these changes, I think will be super important if we actually want to understand how climate change will be affecting disease in the future. And with that, I'd just like to thank a ton of people who helped with all this research that I presented, and I'd be happy to take questions.